Hey guys! Today we're going to go through respiration, and this is based on the OCR specification for A-level biology. So the first stage is glycolysis, and this starts off with a glucose molecule. And the first step is phosphorylation. So this is when two ATP molecules donate a phosphate molecule to the glucose, making hexose bisphosphate. And this is an unstable molecule, so it later breaks up into two triose phosphate molecules. And this stage is called lysis. And then these triose phosphate molecules gain two more phosphate molecules, one each, from inorganic phosphate molecules in the cytoplasm, and this is called phosphorylation. Dehydrogenation then occurs to make two pyruvate molecules, and they're also dephosphorylated to donate the two phosphate molecules to two ATP molecules. So in total, 4 ATP molecules are made here. But because two of them are used up in this stage, the net production of ATP molecules is only 2, and 2 NADH molecules are also made because of the dehydrogenation at this stage. And this is an example of how ATP is made by substrate level phosphorylation.
So this is a link reaction, which is the next reaction after glycolysis. Glycolysis happened in the cytoplasm of the cell, but then pyruvate, which is the last product of glycolysis, is transported into the mitochondrial matrix using a specialised carrier molecule, but it requires ATP. This pyruvate molecule is then dehydrogenated to form another molecule of NADH, and is also decarboxylated, which means one of its carbon groups is taken off, and this forms an acetyl group. The acetyl group binds to coenzyme A, and this coenzyme with the acetyl group goes to the Krebs cycle. The molecule formed at the end is called acetyl coenzyme A.
So this is the next step called the Krebs cycle. So the acetyl coenzyme A that was formed in the previous reaction carries an acetyl group to the Krebs cycle, and this binds to oxaloacetate, a 4-carbon compound. And then this later forms citrate, which is then decarboxylated and dehydrogenated to form a carbon dioxide molecule and an NADH to form a 5-carbon compound. And this is further decarboxylated and dehydrogenated to reform oxaloacetate. But this also creates an ATP molecule by substrate level phosphorylation, and this is phosphorylation that doesn't need an ETC. And FADH2 is only made in this process, and it takes 2 hydrogens, whereas NADH only takes 1. So oxaloacetate is reformed in this process, and then it goes background.
So this is the final process of respiration, and it happens in the intermembranal space. So this is the matrix of the mitochondria, and this is the membrane of the cristae, and this is the intermembranal space here. So at the beginning, all of the NADH and the FADH2 that are made in other processes of photosynthesis arrive at the electron transport chain. They release their electrons and their hydrogens, so they are reoxidized. And these electrons are then passed into these electron acceptors, which are within the phospholipid bilayer. So when they are accepted, they pass along from one electron acceptor to another, oxidizing and reoxidizing them. And so this is the electron transport chain. However, when the electrons are accepted here, they are raised to a higher energy level, and as they go from one to the other, they decrease in energy level, and this energy that they release is used to pump hydrogen ions from the matrix into the intermembranal space. So that doesn't actually require ATP, it's used with the energy released by the electrons. This builds a high proton gradient in the intermembranal space. And so when a high enough proton gradient has built up, the hydrogen ions go down ATPase, which is an enzyme within the phospholipid bilayer, and the energy released from this passing through is used to synthesize ATP. This is one of the instances where ATP is not made by substrate level phosphorylation. Also, the electrons that once they have finished passing through the electron transport chain, they combine with oxygen, which is the final electron acceptor, as well as hydrogens that have been released from ATPase. And this also can mean that hydrogens that have been released from NADH and FADH2 may bind immediately without being pumped into the intermembranous space. And when the hydrogen and the electrons bind to the final electron acceptor, they form water, which is why one of the products of respiration is water, as this is where it is made. Also, we can say that FADH2, another difference from NADH, is that the electrons released by FADH2 bind to the electron transport chain at a later stage.
This is a summary of aerobic respiration, and as you can see, oxygen is required as a final electron acceptor. So without oxygen, oxidative phosphorylation can't occur, so that's where anaerobic respiration happens. So we'll cover that next time. Thanks for watching.
Bye!